Good evening. I hope you can hear me. So, we'll start with the problem sessions for this week. So, this will be an interactive session. We'll look at questions. Whoever knows the answer, or if you know something relevant to the question that's being asked, you can raise your hand and then we'll start discussing. Okay, so the first question that we are looking at is identify one natural source of secologanin which can be used as a replacement of chemically synthesized secologanin. And four different options are given here. Fragaria ananassa berries, Oxycoccus palustris berries, Vaccinium cyanococcus berries, and Symphoricarpus albus berries. You can take a couple of minutes. First, think about what this compound is. Secologanin. And then think about the different kind of berries here. Most of these berries are familiar. It's just that the scientific name is given here. You might have come across these. You can take a couple of minutes and then you can raise your hand if you know the answer. Okay. Does anyone want to try? Sonita, Kritika, or Dipali? Does anyone here know what Secologanon is? Okay, so cephalogamin is a iridoid glycoside. It is a moiety that is attached covalently to glucose that is specifically present in snowberry plant. And here, option D, Symphoricarpus albus. This is snowberry. Now, this secologamin, it has been studied and has been found to bind to one of these proteins called GSK3 beta protein and has also shown to improve memory and cognitive impairment. So this is just a memory based question. We'll move on to the next question. Select the terpenoid indole alkaloid that is synthesized inside the transgenic yeast cells upon expression of strictocidin glucosidase from CROCS. Four different options are given here: cathinamide, tabersonin, vinblastin, catharanthine. You can take a couple of minutes and then start with explaining what CROCS is, what this enzyme, strictocidin glucosidase, would do, what are transgenic yeast cells. We can discuss it in that manner. So you can take a couple of minutes, and whoever knows the answer can raise your hand and unmute and then talk. Okay. Does anyone want to try answering? So we have the pathway here, starting from shikimate pathway, which gives us tryptamine. What we are interested in is this step, where strictocidin glucosidase is being expressed. Now, this pathway is naturally present in Catharanthus roseus. And now, because this enzyme that codes for strictocidin glucosidase from Catharanthus roseus is being expressed in yeast cells, this process is called transgenic or heterologous. So, this strictocidin glucosidase acts on strictocidin here. So, that step is shown here. I hope this is clear. Strictocidin glucosidase is this SGD enzyme that converts strictocidin to a strictocidin A glycon. So here what you see as OGLC is a glucose moiety attached to the oxygen atom here. This is covalently linked. Now moving to the question, which of these is synthesized inside this transgenic yeast if this particular enzyme is being expressed? So what we need to see is what are all the possible downstream products that will be synthesized if this strictocidin glucosidase is being expressed. So we have these many different options, among which we have tabersonin, we also have catranthine. So we have tabersonin and catranthine. So how do we decide among these two? Does anyone want to uh, answer for this? How do we decide between tabersonin and catranthine? Because both seem to be downstream of the strictocidin A glycon. So which of these should we choose? So the way to choose here, yeah, can you hear me? Okay. Huh. So the way to choose here is that which of these is like the actual direct precursor. And if we look at this, these this looks like a direct pathway compared to this, which is why we will go with tabersonin for this particular question. The next question is identify the false statement regarding terpenoid indole alkaloid biosynthesis from the following. And four different options are given here. Hairy root cultures of C. roseus under dark conditions detected tabersonin. Hairy root cultures of C. roseus under light turned green in color. Hairy root cultures of C. roseus under light produced ajmalicin while tabersonin reduced. Or hairy root cultures of C. roseus under light produced windolin while tabersonin reduced. So to answer this question, there are a couple of things that you need to think about. One is this process of etiolation, where you grow cells or grow plants in the presence and absence of light, and how the presence or the absence of light would dictate production of specific metabolites or diversion towards another pathway. Think about these two things. We will also look at the pathway that was shown for the previous question to be able to see if any of the conditions and at the pathway level, which would be the appropriate answer here. So you can take a couple of minutes and then raise your hand and then we'll discuss. Okay, does anyone want to try answering this question? The question is about how, uh, under different conditions of light and darkness, this terpene indole alkaloid biosynthesis would produce different compounds. Uh, so we want to know which of these is false. Does anyone want to try? Deepali, Shrikant, Swati? Okay. So we'll look at each of the options given here. So the first thing that we're going to look at is about Tabor-Sonnen production under dark conditions. So Tabor-Sonnen, it is in between windolin and windblastin production. It is one of the compounds that is needed for windblastin production. So if we look at this, Tabor-Sonnen gives rise to windolin, which is needed for windblastin. production. Now, what is observed is that this can be produced only under dark conditions, and if light is given, that would alter the metabolism towards other components. So, this is a true option. The second option is hairy root cultures of cirrhosis under light turned green in color. So, this can be explained by chlorophyll biosynthesis, which is why this is done in the absence of light. Option C is that hairy root cultures of cirrhosis under light produced ajmalicin while tabersonin reduced. Now, what we need to know here is whether ajmalicin is a part of this indole alkaloids. So what we are looking at is terpenoid indole alkaloids, and they are talking about shift in the metabolism in the presence of light. But ajmalicin is a part of the different branch of this indole alkaloid and is not really affected by light. Like we saw in option A, light will only uh, alter the metabolism from producing tabersonin to windolin, but not towards ajmalicin. So this is the wrong option. Also, there is one more correct option that we need to be aware of, which is hairy root cultures of C. roseus under light produced windolin while tabersonin reduced. So here they say that light will activate the enzymes that are required for converting tabersonin to windolin. Now, like we saw earlier, if tabersonin is converted to windolin, of course tabersonin's levels will be reduced, which is obvious, and that is given in option D. So this is also true. So the only thing that falls in this question is that it will produce ajmalicin, but that is not true because ajmalicin is a part of a different pathway. Okay. The next question is select the correct transcription factor from the following options that leads to tryptocidin synthase activation upon jasmonic acid treatment. And four different transcription factors are given here. Option A TAF, option B ORCA3, option C ROS1, option D Ninja. You can take a couple of minutes and then think about, okay, the first thing that you can do is try to give the full form of each of these different transcription factors. Each of these different transcription factors belong to different families associated with specific responses. These transcription factors are generally associated for gene expression with respect to a particular environmental stimuli. So first we look at what each of these transcription factors deals with. Then we will try to shortlist which of these is essential for or uh, acts in accordance to jasmonic acid treatment. So whoever knows the answer can raise your hands after a couple of
The,
minutes.
the
After a couple,
options are not
the options are not. Sorry.
visible.
Wait, are
They're
you
not
able to
visible.
see my slide? Are you able to see my slide? Are the options visible now?
Yes. Yes.
Okay, cool. You can take a couple of minutes and then first come up with the full form for each of these transcription factors. Then we'll go through the function of each of these. Okay. Yeah, Swati, you can start with the options. You can first tell the full form of each of the options. We'll take it from there.
Full forms I forgot. Sorry.
Okay.
I
L-
think the
uh,
correct transcription factors hello.
yeah, yeah, we can hear
OR,
you. Or
OR,
K3, is it?
or ORCA3.
Okay. Are you okay? Leave the full forms, but do you know what the other transcription factors would do? It's okay if you don't know the full form. Some of it it's easy to remember, like Ross. Okay, we'll go through the different options and then come up with the right answer. Okay, so TAF is okay. So in transcription, we have something called as Primno box. We have okay. Transcription is the process where you have mRNA that is being produced from DNA. This mRNA is produced after the promoter is being identified. mRNA is produced after promoter is being identified, and RNA polymerase can uh, allow for transcription. Now, there are some regions called the Tata box, and some proteins associate or bind with the Tata box. The transcription factors that bind with this Tata box binding proteins are called TAF. These are general transcription factors. These are not uh, expressed under specific conditions. Okay. The next is ROS one. ROS. Does anyone know the full form of ROS here? Okay, so ROS is like you see here, it's a DNA demethylase, and it will. So some transcription factors can specifically bind to methylated DNA. This ROS one will demethylate methylated DNA and prevent the transcriptional gene silencing. So this is involved in DNA repair. So you can think of conditions, cellular conditions where there is reactive oxygen species. That is where this would function. The third one is ninja. So, does anyone here know the full form of ninja? Okay. So, ninja is a novel inhibitor of jasmonic acid. It will negatively regulate this pathway and it will not activate the pathway. Yeah, Pujita. Sorry, I could see your answer only now. Yeah, reactive oxygen species. Good. Ninja is a repressor protein, but it is not a activating one. So the question is about correct transcription factor that would activate upon jasmonic acid treatment. Now for this, we have this specific transcription factor that is responsive to jasmonic acid alone. It is called ORCA3. Its full form is octadecanoic responsive catharanthus AP2 or ERF domain protein 3. So this would activate the expression of strictosylin synthase, which in response to jasmonic acid signaling. And an additional information is it belongs to this transcription factor family called AP2 or ERF. Yeah. The next question is select the indole alkaloid that gets accumulated in etiolated seedlings of C. roseus. You can take a couple of minutes and then first tell us what etiolated seedlings are. Then we'll go to the specific indole alkaloid that gets accumulated in these etiolated seedlings. Okay. Anyone tell me what etiolated seedlings are? Is it any specific growth condition? Yeah, yeah, you can unmute. Yes, Hati.
So, alkaloid gets accumulated in the seedling.
Sorry, sorry.
Um,
Can you repeat?
think, uh, I think the option C, catharanthine is the from tryptophan, tryptamine is produced, and then by streptosidase, and then catharanthine is produced.
Yeah, but we'll first go through what etiolated seedlings are because that is important for this particular question. Do you know what etiolated seedlings are? We didn't use the term earlier, but we discussed the concept earlier in a previous question.
Estimated siblings, I was not there, I think. That's right.
Sorry, I can't hear you. Etiolated seedlings are?
No, if you discussed in the earlier question, I was not there in that
We didn't use the. Oh, right. Okay, okay. So earlier we were looking at this specific question under light and dark conditions. Uh, which of these statements are true for the production of terpene alkaloid, terpene indole alkaloid in cirrhosis under light and dark conditions? That was the question that we were looking at earlier. Now, etiolated seedlings are those that are cultured in the absence of light. Etiolation is the process where you culture a plant in the absence of light, so basically in darkness. Now, in Catharanthus roseus, there is an observed shift in the metabolism and in the expression levels of enzymes in the presence and in the absence of light. That is established. Now, what you ask is which of these compounds get accumulated in etiolated seedlings. So, you need to know that in cirrhosis, in the presence of light and in the absence of light, different compounds are produced. There's a different distribution of the compounds. So in darkness, which of these compounds is produced? That's the question. Earlier, we saw from the pathway that if cycloserine is produced, that would give rise to tabersonin, it would give rise to vindolin, vinblastin, catharanthin, and all of these. So technically, all these options are present. But the question is specific for in the presence of in the absence of light. So think about it. Except for ajmalism. So what is said is if we want windowlin, which is this at this step here, where you have the enzymes DAT, D4H, NMT, and so on. If you want this metabolite called windowlin, that will require light. But if it is in dark, this windowlin will not be produced, which means that tabersonin, which is the precursor for this, will get accumulated. Okay, was it clear?
So, a windowlin, it depends on light. What
Yeah.
was the question?
The question is which of these get accumulated in etiolated seedlings? So that's where we started.
In absence of light.
Yeah, etiolated means growing in dark conditions, in the absence of light. So these enzymes have a specific nature where they get expressed in the presence and absence of light. That is how you have a shift in metabolism in plants.
Then what is the correct answer here?
So the correct answer here is tabersonin. So the point
Tabersonin.
is you have windowlin being produced in the presence of light. If there is no light, the precursor to windowlin will just get accumulated. So windowlin is produced from tabersonin. That's what we saw here. You have tabersonin here. After a series of steps, you have windowlin here. But this windowlin needs light. If there is no light, the enzymes will not be active. If the enzymes are not active, then the precursor is going to get accumulated. That is how we say that tabersonin is the one that gets accumulated. It is similar to how in other pathways where we have gene deletion that will prevent the production of undesired metabolites and accumulate a desired metabolite. Similarly, here, instead of deletion, uh, we just exploit this natural system where light is essential for enzymatic activity or enzyme expression. Okay. So the next question is identify the compound that is not an indole alkaloid from the options provided. And four different options are given here: catenamine, benzaldehyde, ajmalasin, and stemadadenine. You can take a couple of minutes, think through the question, and then unmute and then answer. Okay? Okay. Does anyone want to answer? It's very evident from the name itself. Catenamine, benzaldehyde, ajmalasin, and stemadadenine. Yes, Pati.
Cathinamine, agmalicin, all are indole alkaloids.
Right.
Benzaldehyde, I think, is the answer.
Yeah, that's the chemically synthesis. Benzaldehyde, we know that you can't get it from plants, and it's yeah, all of these are aromatic, but this is specifically not a terpene indole alkaloid. That's this indole structure is missing, so that's why it's not an indole alkaloid. Right. The next question is which pathway intermediate of indole alkaloid pathway forms by fusion of tryptamine and secologanin? So the options are strictocidin, reticulin, ajmalicin, and sanguinarin. So here we have in the earlier weeks also discussed about reticulin and sanguinarin. In today's class, we have discussed about strictocidin and ajmalicin. You can take a couple of minutes and then try to think about the chemistry between tryptamine and secologanin. Then we'll discuss the answer. You can take a couple of minutes and then unmute. Yeah, you can unmute.
Yeah, that's about biosynthetic pathway.
Huh.
So the answer will be and secologanin, which are indole alkaloid. It is streptocidin, option A.
Huh. Do you
By
remember
SPR
what?
to
Sorry. Go ahead. Sorry.
yeah, by the enzyme SPR to streptocidin.
Huh.
Uh, it is
Do you remember what
streptocidin.
reticulin and sanguinarin are? We had discussed this earlier in the previous weeks.
Uh, Equivalent we have discussed sanguinary also.
Yeah. Do you remember which classification of are these alkaloids?
Yes, yes. These are the alkaloids, sanguinary and the reticulin.
Huh.
the reticulin to thebaine, and to codeine, morphine.
Right. So we have discussed about these isoquinoline alkaloids, benzyl isoquinoline alkaloids, BIA. That's the pathway that we were
And
discussing
then
earlier.
benzyl isoprene.
Yeah.
Yes.
So reticulin and sanguinarine belongs to that, right? Uh, Correct. The next one that we were looking at was like you mentioned, strictocidin, for which the pathway was also shown. So strictocidin is here. Tryptamine is produced from the shikimate pathway from tryptophan, which undergoes a decarboxylation reaction using tryptophan decarboxylase, and then we get tryptamine. Now, this tryptamine would act along with secologanid to give us strictocidin. That's what we asked for in the question as well. And yeah, so strictocidin is the right answer. So what in the answer we have that reticulin is a precursor for benzyl isoquinoline alkaloid, not indole alkaloid, which is what we were discussing. Sanguinarine is a benzophenanthridine alkaloid, again not an indole alkaloid. Ajumalsin, it's an indole alkaloid pathway, but not formed from tryptamine and secalogalin. Whereas strictocidin is the one that is formed by enzymatic condensation of tryptamine and secaloganid. And the enzyme that is needed here is streptocidin synthase. Uh, and we saw earlier in this pathway also that how it can act as a precursor for synthesis of many of these compounds, like vinblastin, vincristin, azimalsin. Reserpin we didn't see. The next question is, which of the following is a dimeric indole alkaloid? Options are vindolin, vinblastin, catharanthin, and azimalsin. So before we go to the answer, first can someone like you can take a couple of minutes, think through, and then tell what dimeric indole alkaloids are. How are they different from normal alkaloids? Okay. Excuse me. Yeah. Does anyone want to answer what dimeric indole alkaloids are? Swati or Deepali? What are dimeric indole alkaloids? Okay. So dimeric means there are two molecules. Now, earlier we saw the pathway where catharanthine, vinblastin, and vindolin were produced. We'll look at the pathway again. So, the compounds that we are talking are vindolin, catharanthine, and vinblastin. Till this point, we see that there is one molecule with these many rings, and the molecule with these many rings. Whereas for vinblastin alone, what we observe is that this vindolin and catharanthine are chemically coupled to produce vinblastin. And this coupling happens at this position. So, because this is a chemical coupling of two molecules, this is called as dimeric. That is why among the options given here, vinblastin is the dimeric molecule. Okay. Another representation of the dimeric indole alkaloid vinblastin is shown here. The next question is, which two monomeric indole alkaloids used to form the dimeric indole alkaloid vinblastin? Again, it's a repetition of the previous question only in a different format. They ask about cathinamide, vinblastin, uh, catharanthine and vinblastin, catharanthine and vindolin, catharanthine and vincristine. So, like we saw in the earlier question, catharanthine and vindolin is what gives us vinblastin. So, option C is the right answer here. Yeah, one thing not mentioning here is, like vinblastin, we have something called as vincristine. Vincristine itself is a dimer. It can be linked to catharanthine. The last question for today's class is, mark the correct biosynthetic pathway for serpentine formation from strictocidin. The options are, okay, strictocidin to strictocidin aglycone to cathinamine to admalcin to serpentine. And we have similar options. So you can take a couple of minutes, think through the options, and then tell us what will the possible pathway be. Okay. Okay. So the pathway for formation of streptococcus, the formation of serpentine from streptococcus. Yeah, you can unmute.
I think the one is the correct answer.
Option A, is it?
In option A, the streptocodon to streptocodon glycon, and then the remaining
Yeah, option A.
xanthan.
Yeah, yeah, you're right. Option A is correct. So,
I just wanted to ask you the
huh.
previous question. I missed out the
Oh,
dimeric. Which
right,
one is
right.
that answer?
Okay. Do you know what dimeric alkaloid are? We were discussing that.
No, I don't know. Yeah.
Okay. So, if you look at the structure of these molecules in the pathway, I'll show the pathway, I'll zoom in. All of these have rings, right? But you have all these rings directly connected in the ring pattern itself. When two of these compounds are covalently linked, there is windolin, which is one ringed compound. There is catharanthin, which is another ringed compound. When these are chemically coupled, you get two sets of rings that are joined by a bond. This is called as dimeric. Okay, so two different molecules that are connected. That is a dimer. So the dimer means two here. And the question here is about dimeric alkaloids, right? So if you look at the options, they ask for windolin, windblastin, catharanthin, and indolcin. We earlier know windolin and catharanthin are monomeric, and windblastin is the only dimeric terpene indole alkaloid. You see here, there is this one ring structure and the other ring structure, and both are linked together. Was that clear? Oh, okay, cool. Okay, so with this, we come to the end of this week's class. If there are any questions, you can ask me. Swati, you were not here for the first few questions, right? Do you remember which all questions you were not here for?
First two, three questions I was not there.
Okay, we'll just look at the questions. You can take a couple of minutes, think through, so that it will benefit you. So the first question is about natural source for secologanin, which can be used as a replacement for chemically synthesized secologanin. So four different options are given here. All of these are berries. You might be familiar with these. One is a strawberry, one is a snowberry, and so on. So you can take a couple of minutes, think through which of these berries would have secologanin. Okay? Yeah, yeah, you can unmute.
I think the snowberry, that Symphoricarpus albus berries,
Yeah. So
the
yeah,
option.
that's the right answer. That's the only one that has secologanin. And this again is similar to one of the compounds that we saw here, strictosudin, in a way that this is also a glycoside. So, glycoside means it has a glucose moiety attached to it. So, seculagaride is also a glucoside, glycoside. Glycoside and glucoside are interchangeable, and this is commonly found in strawberries. Okay. The next question is select the terpenoid indole alkaloid that is synthesized inside transgenic yeast cells upon expression of strictosudin glucosidase from Catharanthus roseus. So we have strictosudin glucosidase from a plant that is being expressed in yeast cells. That is why it is called transgenic. It is not normally present in yeast cells. Now, if we do that, which of these alkaloids will be synthesized? That's the question. You can take a couple of minutes, think how the pathway would like flow through when you have strictocidin being acted upon by strictocidin glucosidase. That would give you something called as strictocidin aglycone. So from strictocidin aglycone, what are all the possible compounds that can be produced?
Uh, Catenamin, I'm not sure.
Okay, I'll show the pathway. Do you think, okay, what is cathepsin? From the name, you should be able to get a slight idea because earlier you were even mentioning about something called as tryptamine, right? What is tryptamine?
From tryptophan to histamine,
Yeah. So it's
then
a decarboxylated compound. Can an amine act as an indole alkaloid?